let's take a moment to talk about some of the genes, specifically along methylation. So there's a lot of hype around the MTHFR genes, specifically the C677T gene. So even if we have a gene mutation there, say 1 or 2 mutations, one mutation would be a heterozygous gene mutation, 2 is a homozygous, we can still convert folic acid to methylfolate. So that gene mutation involves doing just that. When somebody has a gene mutation there, it just means that it's not working efficiently. It doesn't mean necessarily you have to stop eating all things with folic acid. It's just not properly converting it in the body. The other mutation is the A1298C. Now, this one, even though it has the MTHFR in front of it, it really doesn't have anything to do with converting methylfolate. So it's part of a different part of the methylation map. So if you look on a methylation map, you'll see MTHFR and you'll see the other gene mutations like COMT, which I'm going to hit on here in just a moment as well. So this one involves BH4. We use BH4 to get rid of ammonia in the body. We also use BH4 to create neurotransmitters. When you have a mutation on A1298C, your body has a hard time with this. So it struggles with removing ammonia in the body. It can struggle with creating neurotransmitters. But the big thing is really the ammonia there. So when we have mutations, I actually have a mutation on that gene as well. And I have issues with removing ammonia inside my body because of that. So that can impair neurotransmitters, which is our serotonin and our dopamine, which can then cause mood issues, ADHD, other symptoms as well because of that. Now, with these genes, we can't just look at the gene mutation. It doesn't necessarily mean that just because we have a mutation on C677T, that mutation is active and turned on. We have to correlate it with homocysteine levels. And we can't just stop there either. We have to also look at COMT, because the COMT mutation deals with being able to handle methyl donors. And methylfolate is a methyl donor. Methyl B12, also another methyl donor. So if you have mutations and it's an active mutation on the COMT gene, you're not going to handle a lot of methyl donors. And how we correlate and know that if that mutation is active, we look at our dopamine levels, because that's what that affects. And if there is no active mutation on C677T, then we need to look at the CBS mutation, if there is a CBS mutation, because it could be active. And that one can be a tougher one to turn off. So when there is no active mutation, we don't need the support. So if there is no active MTHF mutation, we don't need the methylfolate then. That would be counterproductive. We need to next look at MTR, MTRR, and how our body's doing with B12, and then BHMT. If you give a mouse a cookie, it wants a glass of milk, kind of thing. So this is one big cycle. There's not just little bits. We can't just base off of a couple test numbers. It's awesome if you're looking at your genes and you're wanting to know more information on that, but we have to look at the body as a whole, and we have to look at all of the test numbers. If you have any questions, ask me. I'm always here to help.